Okay, so here is a combined lecture. This is going to go through protein structure and function together, and we'll go over all the main points that the students need to know for their class for the molecular and cellular biology. Okay, so this one is covering basically those two chapters from Alberts or Bergstrom that go over protein structure and function. So what are we going to focus on? We're going to focus on how to build a protein. We'll then talk about once you have a protein, how does it get into its proper structure, what type of folds it has, and how they're maintained. We'll talk about once the protein is built, what are the different domains, motifs, and folds in the protein structure that make it possible for it to do the work that it needs to perform. We are also going to talk about what are some chemical modifications that happen on proteins post-translation and how changes in these chemical modifications or the structure can lead to disease. And finally, we are going to talk a little bit about the different types of work that proteins perform in our body and how they're controlled. Okay, so let's get started. So let's start off by looking at what are the building blocks of proteins and how they're built. So the building blocks of proteins are amino acids. Amino acids have a central alpha carbon. They have an amine group on one side, a carboxyl group on the other, and then they'll have a hydrogen bond and your some kind of a R chain, side chain, which we call the R group. That's the group that can react with other things, but also that's a group that determines the properties for that given amino acid. These side chains can come in a variety of flavors and types, so they can be different things happening based on what the side chain is made of. It also is going to determine properties regarding the charge of the protein in any or this amino acid in a particular pH or environment that it is in. Now, once you have different amino acids, you can click them together or bind them together through covalent bonds that are called peptide bonds, and we'll go over how they're formed. So in a non-ionized form, you are going to see that there is no charge on this particular amino acid. In its ionized form, you will have a negative charge on your carboxyl and a positive charge on that amine group. So there are amino acids can be found in two isomeric forms, the L form and the D form. They are just mirror images of each other, right? So these are stereoisomers. The alpha carbon is asymmetric. That allows this mirror image isomer to occur. In our body, all the proteins are going to contain exclusively the L amino acid. That's the form that are going to be used to build proteins inside the body. There are 20 major amino acids that are found in organisms, and these are the varieties come from the side chains that are being put on these amino acids in that little slot. And so you have some that are negatively charged, aspartic acid and glutamine, others that are positively charged. Then you have a host of uncharged polar amino acids. All of these would be examples of hydrophilic amino acids, right? Because they're either charged, negative or positive, or they're polar by nature based on that R group, and that allows them to be miscible in water. In addition, there are about equal numbers of non-polar amino acids that are hydrophobic and would not be miscible in water. Now, amino acids can also be written instead of in their full form in these three-letter abbreviated form, or they can be denoted by single alphabet letters as indicated here. While you don't need to know what these are, you should be able to identify that that is, a string of amino acids versus something else, and you should be able to give me the different type of categories of amino acids based on these properties. Now, how are they put together? Two amino acids, to put them together, you will see them in their ionized form. They are going to create that peptide bond by removal of a water molecule, by taking two of the hydrogens from one and the oxygen from the other. This stripping away from water is again an example of dehydration synthesis, which is how macromolecules form. So it's the same process. However, in this case, we just have a special name for it because it's building a protein. It is called a peptide bond. So here you have the peptide bond between two amino acids that you can see, and you have their R groups represented here. The amine group, the peptide bond forms between the amine group of one amino acid and the carboxyl group of the other. So that's why you have this bond right here. So it's between the nitrogen and the carbon. Now, proteins are made of strings of amino acids that have been linked together to form this kind of chain of amino acids at the most basic level. Now, the R group doesn't have to be all on one side. They're actually going to be alternating. So these are the side chains, right, of these amino acids. So you'll have them on opposite sides, and they'll alternate all throughout the polypeptide chain. Um, Now, in this case, you have methionine, aspartic acid, leucine, and tyrosine. All amino acids are going to start, all proteins, not amino acid, all proteins, polypeptide chains are going to start with the methionine, and then we'll continue with other amino acids in tow, and we'll learn about that later on as well. The amino terminal is going to be at this beginning, and the carboxyl terminal will be at the end of the polypeptide chain. Now, let's see what happens once you have that polypeptide chain strung together, ready to go. So the proteins don't normally exist in this kind of linear form. They usually exist in a 3D form, special forms to have special functions. And so we'll talk next about this protein structure. Okay, so there are 4 levels of structure for proteins, and starting with your primary string of amino acids, that's your simplest form, and then quaternary is that final functional form that is going to be able to do more actual work inside the body. So we're going to go through these and talk about what exactly are some main components for each level of order for these protein structures and how it is done. So to begin with, you have your primary structure. Primary structure is just the linear order of amino acids in a polypeptide chain. They are linked together with those polypeptide, with those peptide bonds between the carbon and amine, carboxyl and amino terminal of adjacent amino acids. And it is going to contain your R groups in these kind of alternating manner on one or the other side of the polypeptide chain. This can be figured out if you know the sequence of your gene. Looking at the genetic code, looking at that mRNA, we can predict what amino acids will be strung together, and we can find out that polypeptide chain sequence. However, that doesn't exactly tell you right away what that final form will be, what that structure will be at the end, although we can get ideas about that based on our knowledge of different amino acids and how they like to be, what their properties are, and how they'll be put together. But this primary structure is just the string of amino acids. Next, you have your secondary structure. The secondary structure is where the backbone, so the backbone, when we are talking about the backbone, it just as the backbone of the amino acids, the alpha carbon, the carboxyl, and the amine group without taking into account any of the R groups, okay? So this one is going to start folding up the protein automatically, that long ribbon or the string of amino acids that are stuck together as they're built based on the different properties of amino acid side chains. So let's say there's a hydrophilic one, so it wouldn't bind being outside towards the water or towards the cytoplasmic phase, and others that are hydrophobic would try to kind of, go internal and fold up so that they're protected. And based on those type of things that are happening, you will start to get some basic structures. Two of these structures that happen in this secondary level are alpha helices and beta sheets that we've already talked about a little bit in when we were talking about membrane proteins. So should be familiar to you. So these are the two main parts of the secondary structure in proteins. These are maintained through hydrogen bonding in the side chain. So you can see there in the oxygen and the hydrogen, and maybe, so that it is stabilized in this manner. But that's what it is. So the hydrogen bonds between the side chain, and not the side chains, but the backbone, are what is going to maintain the alpha helices and beta sheets. And there are going to help, these are some local folding patterns, repetitive ones that you see again and again in different proteins. And they're going to help that protein start to fold up in its final structure. So let's look at these a little bit more carefully. The first one is the alpha helices. Again, the side chains are going to be now either towards the outside, and those backbones are going to be interacting with each other and making these hydrogen bonds in between the carbon, hydrogen, oxygen, anything like that, right? And that's what's going to determine how they're going to start to fold up. They have a very particular structure, these alpha helices. They make a particular turn and repetitive manner that I will show you in a second. In beta sheets, again, you are going to start to see hydrogen bondings between adjacent layers of your polypeptide chain, the same chain, not different, not others, the same one folding over. And in this one, they can fold in parallel or anti-parallel manner. But again, the R groups are going to be facing up or down, and the side chains laying parallel to each other are going to have hydrogen bondings between them. So let's start off a little bit looking at alpha helices. And this is, again, now we're going to use an example of a membrane protein that you're comfortable with and you've seen before. Alpha helices basically form these kind of cylindrical coiled structure that is going to allow proteins to cross the lipid bilayer to be, transmembrane proteins. They can be membrane spanning. They don't have to be, they could just be, partially embedded, right? So they could be one or the other side. To span the entire double layer would be about 20 amino acids. To transverse the membrane entirely, 10 would take you to where it is either on the cytosolic side or the extracellular side, or one or the other side of the bilayer. These are formed through hydrophobic side chains interacting with the lipid layer. So these side chains on the outside are the ones that are going to interact with the lipid bilayer. And then you'll have these sparse amino acids that may be facing inside. Those would be your hydrophilic ones. Now, if you have just a single coil structure, most of the time, if it's a transmembrane protein, that's single passed and transmembrane protein, most of these amino acids are going to be hydrophobic, facing outside with their R groups. And inside, because of all the stability between them through hydrogen bonding, you're going to get a rigid cylindrical structure that is anchoring that protein within the membrane. Okay, so it is very important for structural integrity of membrane-bound proteins. Now, these can be left-handed or right-handed helis. It's going to depend on where those proteins are, where the R groups are, how they're twisting based on the other structures around it. And they can be complex or simple. If you have a single pass, you're just going to get a single coil, and it could be left or right-handed. If you have multiple ones of these coming together, they could be forming a more complex structure or even a pore, as we've talked about before. And in this case, each one of these would represent a single coil, right? They're stabilized by the nitrogen-hydrogen groups of one amino acid having hydrogen bonding with the CO group of another that is 4 residues apart, or 4 amino acids apart. This gives it this classical 3.6 amino, so there's a helical turn, a very clear twist every 3.6 amino acids because of the way they're hydrogen bonding together with every fourth amino acid. Now, how do you get them? Remember, we talked about some proteins where they have a hydrophilic side and a hydrophobic, and you have that pore. How do you get there from this simple structure? Because looking at that looks like everything is hydrophobic. While you can have a repeating 7 amino acid sequence where specifically amino acid A and T are hydrophobic and will face towards the hydrophobic side of your area that they are embedded in, and the rest of them are hydrophilic. Now, here is an example of what some of these proteins can do. So just because you have an alpha helix doesn't mean it has to be a membrane protein. They can be in the cytoplasm as well, and they can be extremely important in structural components of your cell, right? So for example, you could have these structural proteins that are coiled to create like a rope-like structure, right? In this case, these specific amino acids, A and D, when they are all hydrogen bonded together, they create this hydrophobic stripe along the length of this polypeptide chain. Now, alone, that's not going to really help because you're going to have a hydrophobic and a hydrophilic side. You have to kind of get in touch with another one of its own so that it can coil with it together along this hydrophobic edge. That's going to create kind of like a zip, like a little sealed structure where like you're putting two tapes together. And that now these hydrophobic amino acids are protected inside, and all these other hydrophilic amino acids are facing towards the outside. In this case, this can be in an aqueous environment, in the cytoplasm, or anywhere else. So coiled structure will contribute to, again, the structural integrity of the protein and can also help maintain structures of cells through them, because these are going to be very stiff and very structurally inert areas of those proteins. Now, beta sheets, again, these are going to be hydrogen bonds between segments of polypeptide chain laying side by side or parallel to each other. They can be in parallel or anti-parallel orientation. So here's an example where a protein has these areas that are going to create these beta sheets linked together with this area that is not part of the beta sheet structure. And in this case, they are laying anti-parallel to each other. Here you have ones where you have single, them all oriented in the same direction, connected with areas that are not part of the beta sheet structure. And again, this would create this little sheet area, right? A little mesh-like area in that protein that is very strong in its bonding. Now, once you have those set, you can imagine that already gets the protein kind of in a 3D form, because their position is going to be very stable where they are, the alpha helices and beta sheets. The rest of the chain around it will now conform to reach the lowest energy state, essentially. So in tertiary structure, you finally get to see that 3D form. And this 3D folding comes from the remaining amino acids that are freely kind of amorphous around these structural domains, and the side chains now having interactions with each other, coming close to each other or to the backbone and creating their own bindings. These can be most of these are going to be hydrogen bonding, but you will also have more stable covalent bonds like disulfide bridges. You can have salt bridges with ionic bonds. You can have hydrophobic interactions when two R groups that don't get along with each other are nearby, and they cause that area to be a certain way so that they can have minimal interaction, stabilizing that shape. So you can have backbone to backbone bonding, you can have backbone to side chain, as well as side chain to side chain. So in tertiary structure is where side chains actually come into play to create that 3D form. Finally, some proteins are fine. That's it. That's their final functional form once they get to their 3D structure in the tertiary form. Other proteins, and actually many proteins, they don't work alone. They work together with another protein of their own type or a different type. When multiple subunits are coming together, so two or more side chains are coming together, polypeptide chains are coming together to create the final functional form. That's what's going to make its quaternary structure. They're going to be stabilized through the same type of interactions as you've seen before. So again, through hydrogen bonding as well as through salt bridges and disulfide bridges. And this is going to give the final functional kind of, structure for these proteins so they can do their complex functions they need to perform. Here you have an example with hemoglobin that is composed of two alpha hemoglobin and two beta hemoglobin molecules coming together. And not only do they come together by themselves, but they also have additional functional groups that are going to be part of that structure to make it actually do the work that it needs to do, which is to recruit oxygen and carry it around the body. So large proteins that you are going to see would often need more than one polypeptide chain subunit to create their final functional form. They can be in dimers. So here is a dimer where the same protein, identical protein, two of them come together, bind together at this interaction point, and then work like that. Or you can have a tetramer where you have different types of non-identical binding sites coming together onto the monomers to create that complex protein structure. Now, what happens? How do they stay in this place, right? What we have found through studies is that if we purify proteins from cells and expose them to high concentration of salt or urea, again, like urea or another type of salt, they will denature and they will go into their primary structure. So they'll be just a string of amino acids. If you remove that urea or salt or stabilize the pH back to normal, what you will see is that the proteins will go back to their original conformation all on their own in many cases. That means that each protein has a particular set form that it will be in. And what we have found is that that form is determined by the lowest energy state that allows that protein molecule to be the most stable. Now, a lot of proteins, like I said, can reform just by themselves. However, other proteins require a little more help. So there are certain proteins that can start to get into their final form all by themselves, but cannot completely finish because they are present in an environment that isn't exactly what they need to be in that final form. Let's say, for example, that it's a protein that needs to work in a lysosome in that highly acidic environment. In the cytoplasm or in the ER, it's not going to have that exact requirement, environment. So it won't be able to get into its final state just on its own because it's not made for that environment to be in that 3D structure. It's actually in its, and not in its correct environment. So what we have are these proteins called chaperones, and their job is to get proteins to fold properly when they're unable to do so in the environment they are in, and kind of get them to be in that place they need to be. So these were originally isolated in bacteria, and there are different ways that they can be found. So I'm going to tell you two different ways that chaperones can work. In one of them, the protein, when it's made, it starts to create the alpha helices beta sheets. It can still do that. It starts to get in a final form, but it doesn't quite get in a perfectly folded form. So chaperone proteins will bind to this partially folded molecule when it is, where as far as it's gotten to, and it will itself folded up the rest of the way to get it into the correctly folded protein form, and then allow it that opportunity to move into the place that where it can be stabilized in this final form. Other times, the proteins need even more, the environment is a very, like the example with the lysosome, it is a very different environment. It cannot fold in that form in a place where it doesn't have that environment at all. In that case, there are chaperone proteins that provide that environment in this kind of chamber form. So in this case, the partially folded proteins will go into these chaperone proteins. They will kind of enclose them. They look like chambers, and they have even a cap-like structure on top of it. And once they have folded it properly, this protein will no longer be stable in here, so they'll release it back outside when the cap dissociates, allowing it to go in where it needs to go. So those are two different ways that chaperones can directly fold it, help it get into the right conformation while it's still where it needs to be, or while it is in its normal environment, or they can create that environment in these little chambers where the proteins go in, and then they get them into the right position and then release them back out. One example of this is a very simple thing, which is disulfide bridges. Disulfide bridges cannot be found in cytoplasmic proteins because just by themselves, since it is a reducing environment. So for that, it has to be with the help of chaperones or in a particular type of extracellular environment that you'll see that happening. Now, proteins will have these structured spaces to create that final 3D form, but that doesn't mean that the entire protein has to be in a very kind of rigid structure. So it's not like a box that it has to be in that perfect box shape all the time. Proteins actually are very amorphous. They have particular structural domains and regions that are needed for it to do its function, for its proper binding. And then they have areas that are more fluid, that are more amorphous, that can move around a little bit more, and that allow the proteins to actually do their job better. So you have proteins, here's an example, that you have these proteins where you can have areas that are very structured based on what they need to do. So here's a tethering domain, for example, in this protein. This tethering domain has to be in that final structural form to be able to clip and tether to this other protein molecule or to this other part of this protein. However, this other area, this long connector, is amorphous. So it can come and open and close as needed under certain conditions, right? Similarly here, to bind, if you have a protein and needs to bind a substrate, it needs to be able to, hold on to it in a particular way to be able to have that good lock and key mechanism. It needs to have a more amorphous, non-structural form so that it can fold around it and interact with it a little bit better and create that structure. And so here you can see this particular protein you have in a long polypeptide chain. You have some structural domains that are important for its binding to each other or to its structure or function. And then you have areas that are going to be a little bit more loosely structured. And this scaffolding portion is going to allow these portions that are structured to come together and create their final reacting form, while the rest of it can be a little bit more fluid. So again, now you see that there are certain parts of the protein that are going to be important for its structure and function, other parts that are going to be a little bit more amorphous. Because of this, you will see that there are different types of structural and functional domains and motifs that can be found within a protein. These are essentially talking about repetitive sequences or structures that you see across all proteins appearing again and again. So domains are example of modular units of sequences that when present will do a particular function or be involved in a particular structure. For example, we talked about a few of them already, these alpha helices, right? And beta folds, these kind of protein folding structures. These are structural domains that when present, when amino acids of a particular type of present in a sequence will create that. Example of functional domains would be when a particular type of sequence or a particular number or unit of modular sequences can create a function. For example, it allows the protein to bind DNA, or it allows the protein to be activated by another protein or phosphorylation or anything like that. Motifs, on the other hand, are shorter sequences. They are again similar to domains, but much smaller. And these are patterns that will have, again, a functional implication. So you can have these helix loop helix, so a small helix followed by a loop, followed by a helix domain or motif that can allow this protein to bind to DNA, for example, right? So things like that. So smaller elements, smaller short sequence patterns that when joined together create a functional group are called motifs. Domains are longer modular units of patterns that when found will either give you a structural domain or a functional domain. So our protein folding are basically what are creating these motifs and domains, right? Based on these amino acid structures. So they are going to be, like the helix bundle, beta barrel. So beta barrel is going to be domains that are created by these beta sheets. That's another example of that. Rosman folds would be another examples of them. Any type of these kind of domains and motifs that come together and are found and again, typically you'll see that some part of it is going to be associated with protein folding. Okay. Okay, so let's go through protein domains a little bit more. These are distinct functional and or structural units in a protein, right? They're going to be important for particular structure or functional interaction, and they'll be important in the final function of the protein. So they can be found in proteins with completely different functions. So you can have two proteins, they both have a DNA binding protein, right? The motif or domain, right? So they both have DNA binding domains. One of them works to as a transcription factor and actually transcribes, helps in transcription of genes, while the other one has a completely different function. Maybe it binds to add histones, to form interaction with histones or to add methylation on it, right? So they can have completely different function while still having that same structural domain that allows it to bind DNA. So here are some examples of domains. You can have, like protein binding domains. You can have subcellular targeting domains that target that protein to the Golgi or the lysosome or the mitochondria or even outside, right? And then you can have catalytic domains that will allow that protein to do some kind of catalysis, some kind of enzymatic function. Now, protein sequence, again, here you see there's a domain, it has some sites. So the protein sequence within this domain is going to now allow it to have certain functional properties. These are going to be regulated through what we call active sites, binding sites. Active sites are places where it can be modified in some way to activate the protein's function. Binding sites allow it to interact with typically other proteins or molecules to have that function performed in some way, shape, or form. So binding DNA, binding another protein, binding another molecule like oxygen, right? Then there are going to be sites that are what we call post-translational modification sites. These are places that will be modified, and typically, active site could be an example of those as well. Where this protein is modified in a way to allow the active site or the binding site to become open and available for function. And then you can have repetitive sequences that can allow that protein to, again, do some other work. So when we classify proteins, we typically classify them based on this sequence and these arrangements of domains and functional groups that are present within it. A family of protein is a group of protein that have some kind of shared function or structure within it. And then domains within that family will be those specific areas that allow it to do whatever that function or structure is that is present as a whole within that family of proteins. And then the sequence features are going to be grouped based on specific sequence characteristics. So here's an example, right? The superfamily over here would be G protein coupled receptors. So that's a particular type, a structural protein, right? It's a particular type of protein with a very specific structure on it. It's a transmembrane protein. It has different types of alpha helix loops, different number of them. G protein coupled receptors will have 7 transmembrane domains specifically. So that's that class, or that's the full family of protein would be G protein coupled receptors. Within it, you can have all kinds of different receptor families. You can have secretin-like receptors. You can have C-AMP related receptors. You can have rhodopsin-like receptors. You can have metabotropic or other functional receptors. So these would be different families of receptors. Between them, if you now break it down even further, and we are going to break down just specifically rhodopsin-like GPCR. So these are light, activated receptors. You'll have opsins, you'll have dopamine receptors, you'll have adenosine receptors, relaxin. Now, some of them are not going to be related to that light activity. Specifically, if you wanted to go to those ones that are light activated, which would be the opsins, you'll have the red sensitive, green sensitive, blue sensitive rhodopsins, right? Based on the sequence that's within these, right? Sequence features, this will just correspond in a particular motif. If that motif contains one set of sequence, it might be red sensitive, another set of sequence would be green sensitive, right? But the domains that make it a G protein coupled receptor would be the same in all of them. So all of them may have a same domain, but then the specific motifs will differ based on the flavor of that protein or the type of function that it performs. So lastly, let's talk about post-translational or chemical modifications of proteins. Now, proteins once built can be chemically modified to become activated or inactivated, and they undergo all kinds of these changes all the time. Each protein can have multiple types of changes that can happen in their various conditions, each one of them making it more active or less active under those conditions. So post-translational modifications specifically relates to phosphorylations, glycosylations, sumoylations, ubiquitinations, any kind of group like that is put on your polypeptide chain at specific amino acid sequences after translation to affect their function at the end of the day. Other types of chemical modifications that can happen are additions of prosthetic groups. This could be like our globin polypeptides binding with a heme molecule, which in turn contains iron, that in turn helps you bind the oxygen. Another example would be proteins that need to be associated with those metal ions for them to be functional. And these could be, again, magnesium, manganese, cobalt, another one, and that allows them to become biologically active. Otherwise, they remain inert. So any type of these post-translational modifications, any prosthetic group that's added or metal ions would be considered these chemical modifications. Now, when proteins are not folded properly, what you will know is that, so here is, for example, a normal prion protein. This particular protein has alpha helices throughout its structure that maintain its normal form and maintain its normal half-life. Now, when you have an infectious prion molecule, protein molecule, what you notice is that some of the alpha helices are fine, but the other two have now converted into these beta sheets because of the type of amino acids that are mutated within them. And that allows them to kind of aggregate, kind of get stuck with each other and create these beta, kind of beta amyloid plaques or like structure, right? Same thing in Alzheimer's, these tau protein tangles, they are formed through similar idea that the protein structure is destabilized, it's different. And now when it gets cleaved, when enzymes destroy this protein, this structure, these little areas are still there and they are not broken down, and they kind of aggregate and create these plaques that can lead to brain degradation and the symptoms with the neurodegenerative effects. So proteins come in a lot of different shapes and sizes depending on the function that they need to perform. They can be globular, fibrous, or membrane bound. Globular proteins, typically, they are going to be the ones that are responsible for a lot of the enzymatic activity within our cells, our metabolic functions. Your fibrous proteins are going to be part of your structural domains and are part of structure and keeping that cytoskeleton and things associated with that. And then the membrane bound may have a little bit of both or whatever the work needs to be done. Some proteins are going to be simple, just by themselves working together. Others are going to be made in a complex where multiple polypeptide chains come together to create the final working molecule. So that brings us to the second part of our lecture, which is to look at some protein functions. We're just looking at them at a more, just a superficial level for now, and then we'll go into details as we go throughout the semester. So protein functions. At the end of the day, if we think about what do proteins do, we'll notice that proteins actually are the worker bees inside our cells, and they are the ones that are doing most of the cellular functions, whether it's enzymatic activity to catalyze chemical reaction and run our metabolism or create new macromolecules, or structural effect, where they are providing structural support to the cells, providing a way for molecules to be taken one place to another, like a roadway structure. They also have transport and motor proteins that can carry molecules from one place to another and generate movement. Then you have storage proteins that can store small molecules of various type within them. And then you have proteins that are involved in signaling and signaling and regulation. These are receptors as well as signal molecules that work together to receive signal from the environment, take them to the cells, and then respond to them. And they respond to them many times through gene regulatory proteins that bind DNA and regulate when certain genes are going to be activated or inactivated. So how do these proteins work? Well, depending on the function they need to perform, they may work through one or more of these ways that we'll talk about. One of them that is very common is basically a change in conformation when the cell receives a signal and transports it to protein in the form of added phosphorylation or activation through another mechanism. This causes a change in the protein shape and either activates it or inhibits its function. Then you have enzyme proteins that are going to be there to get their substrate, allow them to create the product that they need to make through lowering of the activation energy. Then you have allosteric modulations where one of the molecule may bind to the active side of another, allowing regulation of when an enzymatic process should go on and when it should be inhibited. Then you can also have protein-protein interactions where proteins work together to either do a function or to inhibit a function or transmit a signal. Then other proteins, like we said, are going to be dependent on some kind of cofactor, which could be a metal ion, it could be another molecule, it could be a phosphorylation, things like that. Coenzymes, metal ions, these proteins cannot work unless these coenzymes are bound to them or cofactors are bound to them. Finally, proteins are going to facilitate electron transfer and drive redox reactions. And they are going to do so many times based on the environment that they are in. So at certain pH, they may have one function or they may be inert, while at a different pH, they may be changing shape and binding affinity and working in a different manner or having a new function. So when we look at the binding of a protein to another molecule, what we'll notice is that based on that little on the R groups that are contained within that little active site, the bonding is going to be extremely selective. So if you look like, for example, at a glucose versus a galactose molecule, it's so similar. It's just a one tiny shift. Yet the enzymes that will bind the glucose molecule will only bind to glucose molecule during glycolysis and not to galactose molecule. So it's a highly selective binding between a ligand and a protein. These are usually done through non-covalent bonds and through hydrogen bonding or other such interactions. So these are ionic interactions, van der Waals forces, anything like that, and sometimes hydrophobic interactions as well. Now, this selectivity can be best seen in antibodies, right? How highly selective our system is. So in a antibody that our organisms produce as part of their immune response to self versus non-self, these antibodies are built of these four polypeptide chains, two heavy and two light. They have a uniform form or a area that is constant. It's not changing ever. And then on their amine side, they have an area that is highly variable. Depending on every single thing that they're exposed to, that is considered non-self or considered something against the body, they will create variable sequences to bind to that specific antigen only in that specific area only, leading to billions of different antibodies that are possible in our body based on these highly selective binding sites. So that gives you an idea of how specific these bindings have to be. Now, for enzymes, which is a very big class of our proteins, there are 6 classes of enzymes based on the particular function they perform. Hydrolases are going to catalyze hydrolysis. So you can imagine all those macromolecule synthesis that are dehydration synthesis, and then to break them down to release energy is hydrolysis needed to break it apart. So the breaking down molecule using water, when you do breakdown of macromolecules, is catalyzed through hydrolysis or hydrolases. Then you have oxidoreductases. These manage kind of redox reactions inside our body where you're transferring electrons from one molecule to another. We'll see many examples of those. Then you have these things called lyases, which are going to cleave various bonds, but not through oxidation or hydrolysis, but through other means. Then you have transferases that can transfer a functional group from one molecule to the another and allow cellular function and energy transactions between different proteins or different molecules or different parts of the cell. Ligases are going to join two molecules together. And finally, isomerases are going to rearrange atoms within the same molecule without breaking or forming new bonds. So changing a galactose to glucose or changing an L amino acid to the D amino acid, the different isomers, these type of things would be part of these isomerases duties. Now, enzymes specifically are going to bind and alter the substrate molecule. They essentially provide just the right conformation or an environment where the process can occur so that the products can be built, but they don't take part in the process other than that. So they are completely unchanged and they can be recycled and reused as many times as needed while they are there based on their half-life. This enzyme process is encouraged through different ways. One of them is by allowing the substrates to orient properly next to each other the way they need to be so that a reaction can occur. Another way that they can do that is by taking the substrate and rearranging electrons within it so that now it is having these kind of positive and negative sides so that it can have the reaction occur instead of in its inert state. And finally, it may strain and distort in the way that it binds to it so that it is forcing it in that conformation where the reaction can occur. Now, like we said before, sometimes you need additional help, and these help are provided through coenzymes that are organic molecules within the cells, like the heme group, metal ions or inorganic molecules, or prosthetic groups, which are any type of organic or inorganic molecules that can bind to the protein to activate or deactivate it. Finally, we will talk about how proteins are controlled. Proteins can be controlled at various levels throughout their lifetime. They can be controlled through allosteric control, where one molecule binds at some spot on that protein molecule away from the active site, making it unstable so that it cannot have the normal function performed, making it so that the active site is no longer available for the actual molecule to bind to it. Another way is through phosphorylation. So these are kinases that can, or and phosphatases that can add, kinases add phosphate groups to a protein, phosphatases remove them. And through doing that, they can change the functional properties of the protein by changing their binding site. Then you can have ubiquitination. When a protein is ubiquitinated, typically that targets it for degradation. Or modifies its activity greatly to a different way. Finally, you can have these protein-protein interactions where two proteins come together to create, or two or more proteins come together to create a functional molecule and actually perform a function. Now, proteins can have different binding partners, and based on the binding partner they are binding with at a given time, they can have completely different functions. So binding with one protein may make them activate DNA repair pathways, while binding with a different protein may activate apoptosis or program cell death. So the binding partners can very much affect the functionality of a protein. Another thing that can really affect the functionality of the protein is localization. So if a protein is present in the membrane compared to whether it's present in the cytoplasm or the nucleus, they can have completely different roles in each of these locations, depending on the protein that's there and the targets that are present there. In addition, you can have redox regulation through redox reactions to alter the function and active sites of the protein. You can have proteolytic processing, where a portion of the protein is cleaved off and now becomes its own active protein and perform a function. And you can have hormones that can bind to different areas to modulate protein function as well. Proteins can also regulate their own synthesis as well as their activity based on feedback loops that are present between different parts of the pathway. So in metabolic synthesis, just the biosynthetic pathway for various types of protein amino acids, you have areas where they are either positively or negatively regulated to make sure that no amino acid is made in such an abundance that they are not needed, and the cell is not wasting energy creating all these extra product kind of substances that are not needed. So finally, let's walk through how does allosteric regulation and how does phosphorylation work to control this. In an allosteric regulation, the allosteric molecule works as an on-off switch within the cell. So here is an example. Here you have a molecule such as glucose that can bind in this active site, right? And it can either activate it or deactivate it. So let's say that you have a lot of energy available. So you have a lot of ATP, which means that you do not have a lot of ADP present in the cell. So most of the molecules are going to be just not binding to glucose, or if they bind to glucose every now and then, they will still not be active. They will not be able to break it up or do anything with it because they need the ADP bound to it to become active, right? So just having glucose is not enough. You also have to have a need to break it down in order to produce energy. Now, when the cell is low on energy, ATP molecules, you are going to have a lot of ADP in the system because it's already utilized the ATP into ADP. So these ADP molecules are going to come in and bind to this protein molecule. This enzymatic molecule can now bind and stabilize that binding with glucose, leading to glycolysis or any other function that it needs to perform to get the energy needed. So here, this ADP molecule works as a switch to control when it is actually going to do work. So just binding glucose is not enough. It has to do the work as well. It's only going to do it when there is low energy present, low ATP and high ADP, so that it only creates more ATP when it's needed. Phosphorylation can also control energy activity by causing a conformation change. So here it's a little bit different. In this case, you can have protein kinases that add phosphate groups to a protein, causing it to be changed to be active or inactive. It doesn't mean that it has to be activated all the time, although many times it is. It could also be deactivated by that phosphorylation. And then phosphatases would remove that phosphate group and revert it back to whatever function it had normally. So here are two examples where in the first case, the kinase, the phosphorylation is leading it to become on, and the phosphatase is leading it to become off. And in the second state, the kinase is leading the protein to become off, to switch off, and the phosphatase is causing the protein to change shape and become activated again. Here's an example of how this works in a real life system. Here is a protein. This is a protein that we will learn about a lot throughout the semester. It's the tumor suppressor P53. This protein, depending on its binding partner, so you can see there are many different types of binding partners that are being shown in different colors and different types of post-translational modification sites that are being shown. And in each little area, you'll see different functions being represented. So the same P53 can control cell death, can get transactivated and act as a transcription factor, can be stabilized and act to enhance DNA repair pathways, can cause senescence, or can be deacetylated to become inactive. Each section, you will see a different binding partner and different types of changes that are being performed on that protein. Here, you can see its domain structure. So that's your protein, amino terminal, the carboxyl terminal. In the amino terminal, you have the transactivation domain that contains several sites of phosphorylation, acetylations, other things. Then you have the core of the protein where you have additional phosphorylation and acetylation sites, the greens. And then you also have the DNA binding domain that allows it to become a transcription factor. In the C terminal, there are two domains. The first one is the tetramerization domain, and that domain is going to be responsible for it to become a tetramer, so work with 4 molecules together to become an active form. And then the carbohydrate recognition domain is going to help it to recognize specific types of actions so that it can combine with them and do the work that it needs to do. Here again, you see another example of the different modifications that happen. The 50 amino acids in the center DNA binding domain core are the DNA binding site within the core of the protein. Here you have different phosphorylations that can happen. In the tetramerization domain, you have the acetyl groups, you have the ubiquitination groups, you have the phosphorylation, and then in your CRD domain, you have acetylation and phosphorylations. Each one of these will have their own function to control whether the protein is stabilized, activated, or interacting with one or more of these binding partners. Finally, we have these GTP binding proteins that also can act as molecular switches. They work very much like ATP. So GTP can get hydrolyzed by removing the phosphate and going back to the GDP form, which is its inactive form. And then it can be converted back to GTP with the addition of phosphate and turned on in its active form. This can self-hydrolyze to create GDP. That's the good part. It doesn't need a helper. It can just hydrolyze itself to release the phosphate when needed. And it can act like a switch to allow for precise regulation of particular proteins and their function. One example of it working within the cell is when it helps to release tRNA from itself during your translation form. Also, G protein coupled receptors utilize this as well. That's what they are called for. ATP hydrolysis, again, works through removal of one or two phosphates to release, provide functional activity energy to the protein and make it do some work. This is an example with the motor proteins. So the hydrolysis, ATP binding and then hydrolysis allow for this motor protein to move along the cytoskeletal tracts and do the work that it needs to do, whether it is to transport a molecule or a vesicle or just do some other work as well. So that's the stuff that you need to know for proteins. If you have any questions, you can email me or we will talk about it in our class this coming week on Monday. Hope that helped. Take care. Bye-bye.